Welcome to Bioratna. Dear students, I am Chandana Sengupta and our today's topic of discussion is t-RNA, the adapter molecule. Students, during the process of translation, the sequence of nitrogen bases in mRNA, they guide the sequence of amino acids in protein. I repeat, during the process of translation, the sequence of nitrogen bases which are present in mRNA, they guide the sequence of amino acids in the protein. But the question is how? There is no complementarity between these nitrogen bases in the mRNA and the amino acids in protein. But it was known that yes, it is mRNA which is guiding the sequence of amino acids in case of proteins. Because any change in the nitrogen base of mRNA cause subsequent change in the amino acid of the protein, and that's why in the structure of protein. But how it was taking place? When there is A, U, C, G in mRNA, but in proteins there is no nitrogen base. So no complementarity rule can be followed. So how they come in sequence? It was Crick of Watson and Crick who was very clear that as there is no complementarity between these two, then there has to be a third molecule. There has to be a third molecule which acts like an adapter. So on one hand, it reads the codons on mRNA and accordingly it guides the amino acids to take their position. This molecule, it was already discovered at that time and it was known as soluble RNA. It was known by the name of soluble RNA as it is soluble in 1 mole solution of sodium chloride. That's why it was called soluble RNA and its role in translation was not known. When it was known that yes, it is acting as a bridge between the codons and the amino acid, then it was called the adapter molecule. Let us now see the structure of tRNA. Let us first see its general characters. tRNA is a RNA molecule, so it will be showing the general characters of RNA. That is, it is single-stranded. The RNAs, they are single-stranded, but when you see it double-stranded, then the reason is that suppose here there are some nitrogen bases and its complementary nitrogen bases are present at some distance, then this one will fold. This strand will fold upon itself and these two will come together. These two will come close to each other and this part which is not complementary, it will form a loop in between. So wherever you find this loop, this means there are unpaired nitrogen bases. And wherever you find these double strands, that means if here it is A, C, G, C, then on the other strand it is U, G, C and G. Understood? So it is single stranded, but it may form double strand according to the complementary nitrogen bases which is present on the same strand at some distance. Then it is a polymer of nucleotides of A, U, C and G. And the nucleotides, they have 3 components. One is sugar. So this is a pentose sugar and this pentose sugar to its C1 position. 1 is here, 2 is here, 3 is here, 4 is here and 5 carbon, 5th carbon is outside. To this C1 position, the nitrogen base is attached. It may be nucleotide of A or U or C or G. And to the fifth carbon, the phosphate group is attached. To the fifth carbon, the phosphate group is attached. Thus, these are the three components of the nucleotide. Then this phosphate group at C5 position, it forms a diester bond. It forms another ester bond. Here it has formed one ester bond. It will form another ester bond. It will form phosphodiester bond. Here the phosphate group is there. This phosphate, it has formed one ester bond here, and it will form another ester bond with the C3 position of the next nucleotide. Thus, a complete chain will be formed. Now, let us see the more specific characters of tRNA. The tRNAs, they are very small. Actually, they are the smallest of tRNA, mRNA and rRNA. tRNA are the smallest and they have 4S value, S for Svedberg. It is that small. It has less than 100 nucleotides. It has less than 100 nucleotides in it. And it is very specific in its function. Every amino acid has its own tRNA. When we look at its structure, this is the clover leaf. This is called clover leaf structure, or it is called the two-dimensional structure. It was given by Holle in 1964. Actually, tRNA does not look like this clover leaf. In its three-dimensional structure, it looks like inverted L-shape. Inverted L-shaped, and this was given by Clark in 1900. This is 64 and this is 1974. But this two-dimensional structure, it is easy to study. Let us study the different parts of tRNA and how it is coordinating with all the different molecules which participate in translation. This end, its 3' end, towards its 3' end, every tRNA, it has this sequence C, C, A. Look here, this is mRNA having its 5', 3' direction. This is the direction. And this anticodon, it is antiparallel. It is 3' and 5'. And at this CCA end, it is the amino acid acceptor end. Here the specific amino acid gets attached when the tRNA reads the codon on mRNA. We will come to this afterwards. First, it is towards the 3' end, it is the amino acid acceptor end. Let us come this side. One enzyme is required for the attachment of amino acid to the tRNA and to get it charged. That enzyme is called amino acyl tRNA synthetase. This is the loop called DHU, D for di, H for hydro. Dihydrouridine loop where this enzyme it binds. Now come to this side. This is mRNA, and you see that AUG, this codon is present on mRNA. The complementary nitrogen bases present on tRNA, that is UAC, they are called anticodon. So this is codon and anticodon complementarity, and this loop is called anticodon loop. This loop is called anticodon loop. Very important loop. Actually, all are important. Fine. Now, this remaining loop, this loop called pseudouridine loop. This loop called pseudouridine loop, or it is also called ribosome binding loop. So this one tRNA, by its one arm, it is holding amino acid, this side with enzyme, this side with mRNA, and this side with ribosome. This is how it is very nicely coordinating the process. Now when it is, one more thing is that when the codon is here AUG, then the amino acid, the only amino acid the tRNA will carry is methionine. This you already know, AUG codes for methionine. But you need to know the codon and anticodon for two more amino acids, at least these two, as they are given in your NCERT. See this one for serine and this one for tryptophan. Let us first start with serine. On mRNA it is AGU and on tRNA it is UCA. Then it codes for serine. AGU. Fine. Now come to tryptophan. On the mRNA the codon is UAC and on tRNA the anticodon is AUG. That means just reverse of this methionine. The codon anticodon, simply twist it and you get it for tryptophan. And here also AGU na, here see methionine wala AUG hai. The same nitrogen bases are for serine, only the second and third nitrogen base position has been changed. In place of UG, it has become GU. Then it becomes serine. That's it. So this is the structure of tRNA. I hope this video was helpful to you. Then do like and subscribe to stay connected. Do share with your friends. Thank you for watching the video.